Hi. So thanks for the organizers for giving me an opportunity to present our work. And I will present our published paper. And this work was done with my very talented PhD student, Ophir Yaish, on modeling mRNA degradation using deep neural networks. Start with the biological motivation. As we all know, the main dogma in molecular biology is that the messenger RNA is transcribed from DNA. It encodes for a specific protein. And then the messenger RNA is translated into a protein. But RNA, as we all know, is a molecule that degrades quite quickly. For example, the coronavirus vaccine had to be kept in very cold freezers. And in vivo, in the cell, the regulation over the stability of these messenger RNA molecules is mostly controlled by elements found in the 3' UTR, the region that is downstream of the coding sequence, which contains a sequence and regulatory elements that regulate the stability and degradation of the mRNA. So the goal in our study is to find systematic and predictive underlying 3' UTR sequence elements using deep neural networks. Luckily, people and researchers have developed experimental assays to give us lots of data, tell us something about degradation. In this case, we used the data set published in 2017 by Rabbani et al., who was then a postdoc at the Broad Institute. She took 90,000 sequences she derived from the 3' UTR of human genes, and she inserted them into zebrafish embryos and measured degradation through several consecutive time points. So the data, as we see it, computational side, is a list of 90,000 sequences. Each is 110 nucleotide long. And with each sequence, we get the measurements of the mRNA level in different time points. And this was done in two separate biological contexts. One is called A+, that has the poly A tail, and one without the poly A tail, which is denoted as A-. And you see here the different time points that are following fertilization. So it's 1 hour post-fertilization, 2 hours, and so on, and the mRNA levels in log scale. The previous study that generated the data assumed that the degradation happens in a linear rate, meaning it's exponential in the original scale or linear in the log scale. And they took and developed 4 different models, some with early onset, meaning degradation starts immediately, some with late onset. But we found, and I don't have to go over it today, is that many reportants do not fit this linear data. And our motivation was to develop a model that doesn't assume any linear degradation. And I usually say, let the data tell the story, meaning we want to learn from the data what are the degradation dynamics of each mRNA. And one of the motivations that led us to develop a specific model that we developed was measuring the correlation between different time points. As expected, the levels at the first hour are highly correlated with the second hour and so on, and the correlation drops as we go into future time points, right? The lowest correlation has been in the first hour and the 10th hour. But this means that we can think of a model that will enable us to predict the mRNA level at future time points. And this is indeed what we did. We took a model that gets as input the mRNA sequence of the 3' UTR and the initial mRNA level, and then predicts the future time point. So here we see a sequence goes into some pre-processing, which I will go over in detail in the next slide, takes the initial mRNA level, performs some computation using a machine learning model, and then outputs the prediction for future time points. And this is our contribution, which we called deep UTR. We take a sequence, we perform the typical transformation of one-hot encoding, where each nucleotide is now a 4-bit vector with one set for the corresponding nucleotide. We apply a CNN, convolutional or recurrent neural network layer. Then we concatenate the initial mRNA level, we pass it through consecutive fully connected layers, and eventually output what will happen in future time points. So for the convolutional layer, we used 1D convolutions to detect local elements on the sequence level, and then a global max spinning layer. And for the RNN, we used a very popular bidirectional GRU, which takes information both from the 5' to the 3' and from the 3' to the 5'. These are concatenated, and then we added a dropout layer for polarization. And in both cases, we used early stopping using the validation set and a learning rate scheduler. Some technical details over the training and testing. So first of all, we developed a model that is a multitask model, meaning it predicts the mRNA levels in both biological contexts, the A- and the A+, which I remind you are those that include a poly-A tail or don't have the poly-A tail. Also, we made sure that we split the data into train, validation, and test in a way that we don't have homologous sequences. So we generated a graph where each 3' UTR sequence is a node, and we connected by an edge sequences that shared a 20-mer. And then we looked for connected components in the graph. And that way we made sure that between the test, validation, and train, there are no shared 20-mers. In addition, we used sample weighting. Here you see an example of the MSC loss function we used, weighted, where the weight that we choose for each data point is the average read count in 9 time points, meaning we trust more the samples and sequences that have high read count rather than those that have low read count. For hyperparameter tuning, we used a very popular Optuna package in Python. And to improve the robustness and also the prediction performance of the model, we use the random ensemble linearization, which meaning we train 10 different models that differ in the initialization, and in prediction time, we use the average of the models. So for the results, we tested the performance in 2 ways. One, for each of the reporters, each mRNA, we computed the root mean squared error of predicted and measured values. Then for each time point, we computed the Pearson correlation of predicted and measured values over a time point. So one is over a single mRNA and one is over all mRNAs in a single time point. And first, I'm showing you the results over each reporter using the root mean squared error, comparing ourselves to classic models, lasso and random forest, which were developed in previous methods using K-mer features. So here you see that the RNN models perform better than the CNN models, and all deep neural networks models perform better than the classic models. And we also gain by using the multitask version rather than the single task version, meaning predicting for both A- and A+ tasks. As for the Pearson correlation, we see the same trend. Our models based on deep neural networks outperform the classic models, and the RNN models perform better than the CNN models, as well as the multitask better than the single task. Next, we wanted to see how the prediction performance degrades or deteriorates as we move with time. So we computed the RMSE over different time points, starting from the time point that is closest to the initial one, which is the input of the model, to one that is the furthest. And as expected, we see that the performance deteriorates. It's not as good as in the time point that follows the initial one given as input. And what we also see that the outperformance by the deep neural networks is becoming more profound as we go to the further time points, meaning in the more difficult task, we get more gain by using deep neural networks compared to classic methods. We see the same trend when we examine the Pearson correlation, where initially almost all of the models achieve a Pearson correlation of 4.95, but as we go to 10 hours post-fertilization, one of the classic models, random forest, is already below 0.75, while the RNN attains 0.85 in Pearson correlation. Now, in bioinformatics, and I think in general in machine learning, we should always strive and aim to evaluate the performance on independent dataset, not in cross-validation, but on a dataset generated by a different study in a different lab. Luckily, we had such an opportunity in this study we used the RegSys dataset that measured a very similar phenomena of mRNA degradation in zebrafish embryo during zebrafish embryogenesis in the 2nd and 6th hour post-fertilization. And here I'm showing the results comparing using our models trained on a UTR-seq data on this RegSys dataset. Again, we see the outperformance by the deep neural networks, and the multitask RNN is performing the best and achieving almost 0.6 in Pearson correlation. That's also very reassuring to see that a model trained on one dataset is able to predict quite well on an independent dataset from a different lab and a different study. Now, in bioinformatics, we know that we should always look for interpretability. Ask what the model learned and try to discover some biology. In this case, we had 58 validation sequences that were used in the original study in a follow-up experiment. And each one of those had an element embedded in those sequences. So we can ask if our interpretability technique worked well in discovering these embedded elements. For example, here I'm showing you the interpretability results using integrated gradients over the different models, and we see that almost all models pick the poly-U. The models were also able to pick other elements that are destabilizing, and as you see, all models show the effect as a negative effect below the 0 line as a destabilizing as opposed to a stabilizing element. In addition, for the first time, we could also perform a time-wise interpretability analysis, something that was not possible in the previous studies with their models. And here you see that some elements start to kick off at a certain time point or lose their importance in a certain time point. And this is an importance if we're looking for a time-wise analysis. This study also provided an opportunity to benchmark how well we perform interpretability. Since we had the 58 sequences with embedded elements, we knew what was the ground truth in each one of those sequences. So we can ask whether the model was able to identify those elements and then compute an AUC score area under the ROC curve for each one of those elements and report the average. And here we see, as we saw with our eyes, that the CNN models are quite good in interpretability as well as the lasso models. So CNNs are good as lasso in interpretability, much better than RNNs or the random forest. In addition, we wanted to perform a global analysis of the elements that we discover, meaning not just detect elements on a sequence level as we can get with integrated gradients, but also discover common patterns. We used TF-MODISCO that was developed in Anshul Kundage's lab from Stanford, and he discovered the known elements such as poly-U near the microRNA binding site, familial binding site, a rich element, and also new elements that we hope can now be experimentally validated. We also discovered elements that are specific to one of the biological contexts, for example, the A+ or the A-, and one that either changes its role in stabilizing or destabilizing, depending on the context. It will be exciting to discover if these hold an experimental validation. As for future plans, we first thought of improving the prediction by developing time-dependent models, meaning a model that is trained on each time point separately, and also applying temporal constraint, meaning the prediction at the next time point must be smaller than the previous time point if we know that the process is degradation. In addition, we hope to include additional information, such as RNA secondary information that we can get, for example, from RNA-PL fold from the Vienna package, or evolutionary conservation information, where we know that functional elements are probably evolutionarily conserved. In addition, in terms of the interpretability, we want to enable the handling of multiple types of inputs, if we include multiple types, and also discover interactions between different positions. So to summarize my talk, I've shown you that deep neural networks are outperforming conventional methods in predicting mRNA degradation. I've shown you that multitask learning improves prediction performance and also random ensemble generalization. RNNs were better than CNN in the prediction performance, but were worse in interpretability. So if I have to pick one model as the best of both worlds, I would pick the CNNs, convolutional neural networks, that are both good in prediction and interpretability. And last, I've shown you that we discovered and made good progress in interpretability, but I think this is still an open challenge, specifically in genomics, but also generally in the machine learning field. All of the code of the models and the data is available in this GitHub repository, and I want to thank my lab and different funding agencies and you for your attention. So thank you.